Julie, thank you very much for introducing me, but I want to, lest I run out of time at the end, and I almost always do, I want to start by thanking Julie, Desh, Siri, Sabina, everyone who invited me to this conference, even before we had a New Horizons grant, but particularly to CFRI, not just for introducing me to CFRI, but for supporting what I consider to be the euphemism I use is innovative project. Ambitious is the real word. That I really hope will help people. And that's what I'm going to try to describe today. I also want to, I think it's amazing. I want to commend CFRI and Julie and Sabina and Siri in particular for pulling together an amazing effort in a very short amount of time, converting this into a virtual community here in very short order. It's really amazing. So I'm going to talk to you today about a project that aims to improve antibiotic treatment for people with CF. It's largely focused on children, but hopefully by the end of the talk, you'll see that it's really, our hope is that this has broad applicability, and it's a work in progress. I do have one quick disclosure. I have a role on behalf of the CF Foundation as an advisor for researchers who are developing new treatments for CF infections. I start most of my talks this way when I'm talking about sputum. I suspect I don't have to for this audience because people have heard the word before many, many times and probably experience it on a daily basis. But I often feel like it's important for me to describe why anybody would want to spend at least part of their career working on this stuff. And the short answer, as everyone knows, is that lung disease is often the most important or most severe problem that people with CF have. And one of the key drivers of lung disease in people with CF are infections. And we use sputum sort of as our standard sampling of what's going on in people's lungs and their airways. And I'll talk a little bit more later about just how accurate it reflects or depicts the infections in lower airways. And in general, we use what I'll call standard laboratory methods, culture methods, to both diagnose these infections as well as choose antibiotics. And to choose antibiotics, we use this test, which is called antibiotic susceptibility testing. This is just an example. This is actually a plate, a petri dish with agar media in it that's been swabbed with Staphylococcus aureus. This is one of the most, actually the most common CF pathogen in the United States. And on that lawn that's growing of Staph aureus, we put discs of antibiotics, standard antibiotics. And here is one that this staph happens to be resistant to because you don't see this zone of clearing. And it's susceptible to all of these 5 antibiotics. And this is exactly the kind of test that we use to choose antibiotics for most people with CF when they need antibiotics. The problem with this test is that this test, which we use often, does not really reliably allow us to identify antibiotics that are going to work for people with CF. And let me describe what I mean. The way that we diagnose and choose treatments for these infections is by taking the stuff that people cough up, and that's the sputum that I'll talk about the most today. But if people can't cough up, then we lavage it with bronchoscopy. We use a lot of oropharyngeal swabs, increasingly, I'm glad to say, because people are expectorating less and less. And then we take that stuff and we inoculate it in or on selective laboratory media. So what I mean by that is that we have, over the decades, chosen these media to be able to grow the bugs that we think are the important ones, but to exclude the ones we think are not important and we think are contaminants. We then allow those standard pathogens to grow up over a couple of days on a new plate. And we pick just a couple of visually distinct isolates of each of the pathogens on those plates, reculture them. And here is another susceptibility test. This is actually Pseudomonas here. This is a lawn of Pseudomonas on this plate. And each one of these strips is impregnated with a gradient of increasing concentrations of antibiotic. And you can see again that there are variable susceptibilities as shown by this zone of clearance. So based on these in vitro, and by that I mean laboratory susceptibilities of a few isolates of just a few pathogens, we diagnose these infections and we pick antibiotics for our patients and hope for the best. And luckily, gladly, a couple of decades ago, some very wise people started to take stock of this approach, which had been refined over decades and decades, and asked, how's it working? How accurate is it in diagnosing these infections and showing us what's making our patients sick, but also in showing us what's going to make them better? And I have a couple of asides. One of them is the methods I'm going to talk about today focus largely on bacteria. I put the slide in last night because of the talks yesterday. I think I should have put this in because we don't, at least at this point, while our standard lab methods do identify molds and yeasts, we don't focus on them quite as much. We really focus mostly on bacteria, and we rarely focus on viruses or phage, but I expect that both of these are going to change over the coming years. But for these reasons, this talk is largely going to focus on bacteria because of what we consider to be an outsized role in making people sick. And again, that could change. So we'll keep these pathogens in mind for the future. So this is the standard depiction of CF infections that is provided by these kinds of methods. You may have seen this before. This is a graph that's put out by the CF Foundation from their patient registry data every year. And it starts a lot of CF microbiology talks. And this shows you 1 year's worth of data, 2018, and it shows the proportion of people with CF in the United States who had their cultures positive for each of these 6 standard pathogens. These are the most common bacteria that we think are important for CF infections in one subtype, MRSA here. And it's broken down by age, so you can sort of see how these infections change over age or differ. And I'll just point out the 2 most common organisms that we culture. They're Staphylococcus aureus here, most common in kids, Pseudomonas aeruginosa, more common in adults, because I'll talk about them later. So these 6 species, plus a couple that aren't on here because they're not common, like non-tuberculous mycobacteria, these are what we call the traditional pathogens. And these are what are usually identified by our clinical labs. And there are usual targets for antibiotic treatments when we decide people are sick enough to need them. And so again, people have thought about just how well this depiction works for helping us understand what's making people sick and what will make them better. And the short answer is it's not very good. It's disappointing. So for example, a number of studies have looked, really starting almost 20 years ago, 17 years ago, at the relationship between these in vitro susceptibility tests and whether somebody will get better with the antibiotic we choose based on them, and the relationship is quite poor. In fact, there was just a paper published about a week ago that reinforced this. That is, these tests, the in vitro susceptibilities, do not allow us to reliably select an effective antibiotic for anybody. In addition, a number of studies have looked at the relationship between how the density, how much of these pathogens somebody has in their sputum, for example, during an antibiotic treatment trial, and changes in that density and how sick somebody is. And again, that relationship's quite poor. For example, people can have an increase in their sputum Pseudomonas or staph and still get better in terms of lung function or symptoms. So that's confusing. And just to make matters worse, for those of us who take care of young children with CF, studies have shown that a good fraction, at least a fifth, sometimes more, of young children with CF who are sick enough to come to the hospital needing IV antibiotics don't have any pathogens, traditional pathogens identified on their cultures, which makes it very hard to know what's making them sick and how to treat them. So many of us have taken a look at this situation and thought, well, that's not good enough. We really need to do better. And before I talk about our idea for trying to get that, make things better, I wanted to describe how we got here. And it has to do with an amazing success, a real revolution in medicine that was led by lots of people, men and women, but these are the 2 men who are usually identified as the leaders. This is a German named Robert Koch, and a Frenchman named Louis Pasteur. And they, with their colleagues in the late 1800s to early 1900s, developed a set of extremely useful concepts and tools that allowed us to conquer what was then some of the scariest infections that had ever afflicted people. And in general, we now call those acute or classical infections. They were scary because they were often fatal if not treated quickly. And they generally involved the introduction of a new pathogen to what was a sterile space previously, usually a liquid. So for example, this was sepsis in the blood, or infection of the cerebral spinal fluid in meningitis, or the infection of the epithelial lining fluid in pneumonia. These are places where there are usually very, very few or no pathogens or bacteria at all, or microbes at all. And when the bug gets in there, suspended, free swimming in a liquid, as we have in our laboratory tests, it can grow really rapidly. And under those conditions, there's really only one bug. And under those conditions, antibiotics generally work pretty well. So the goal of these methods that these people developed was to isolate and grow that one pathogen, the pathogen that's causing the disease, and tease it apart and figure out what kills it. And these methods then, which usually include rich liquids to grow that one pathogen in isolation, really were developed to specifically reflect those kinds of infections. And they work very, very well. They allow us to often eradicate these infections and treat them before patients get really sick. And so let's consider how CF infections are depicted or reflected by these methods. But to get there, it's helpful to consider how the CF airway becomes infected, at least as far as we think. So under normal conditions, we all inhale tens of thousands of liters of air per day, and all of the stuff that that air has suspended in it, which include a lot of toxins and particles such as pollen, but a lot of bacteria and other microbes as well, as we've learned a lot about in recent months, unfortunately. And as we inhale those microbes, a lot of them impact on the upper airway, the nose, the mouth, the throat, which of course have their own microbes as well. So that column of air loses some microbes, picks others up. And most of those, again, continue to deposit on these increasingly small or decreasingly sized airways as you get to the lung. And in fact, by the time the air gets to the lung, there are very, very few and often no microbes at all. And the airways, all of these airways, secrete a very thin layer of mucus under normal conditions that acts as a vehicle to trap those particles. And then through a variety of methods, we move it out throughout the day and keep our airways clean. And the problem in CF is depicted in this really helpful, I'm sure it's very accurate diagram here. This is supposed to be a cutaway of a healthy human airway. This is 1 millimeter in diameter, if you can imagine that. And this helpful arrow shows you the very thin layer of mucus you can imagine is here under normal conditions. But the problem in CF, just to simplify a very complicated issue, is that these secretions are just thick. It's a maddeningly simple problem that's difficult to remedy. And so these secretions can't be moved out under normal conditions. And they continue to sit there with all of the stuff they've trapped, including microbes, some of which really like these conditions and they thrive, including the bacteria that I just showed you. And so you end up with these cycles of infection and inflammation and damage to the airways that lead to CF lung disease. So the correlates of all of this, and what distinguishes these from acute infections, is that the infection is not growing rapidly in a liquid. It's trapped in a gel, in this mucus. And in fact, that gel is highly heterogeneous. It varies a lot from place to place. And so the infection doesn't eradicate, even though the antibiotics we use tend to work just fine against those bugs in the test tube. And these characteristics are very unlike the acute infections that our tools were developed for. A lot of these bugs actually grow really slowly, and they're trapped in this gel, not free swimming. This gel also has gradients of oxygen. There are some places where oxygen is very low, and that makes the bugs grow slowly. Antibiotics don't work under those conditions. Sometimes nutrients are limiting, sometimes they're very replete, depending on where you are. And that's perhaps one of the reasons antibiotics don't kill all of the bacteria present, but there are lots of potential reasons. And so all of these characteristics are more like what we increasingly call chronic infections, with their defining characteristic being that they just don't eradicate with antibiotics that do work in the test tube. And in fact, they don't rapidly kill people, but over decades and years and decades, they cause persistent and then progressive disease and symptoms despite our treating them with antibiotics that we think should work. That is a chronic infection, and CF really reflects that, exactly that definition. And you can get some clues as to why they're so difficult to treat from thinking about other characteristics that bind together all of the chronic infections that we know about. And the name of the game there, one of those defining characteristics is diversity. CF infections are diverse in many ways. There are several levels of diversity, one of which is diversity in the standard pathogens. If we just look, for example, at Pseudomonas or Staphylococcus or the other standard pathogens, if you were to look at an acute infection with these organisms, like a pneumonia, which is growing rapidly, and you took one cultured isolate of that infecting population, it would reflect the whole infecting population. It's growing so rapidly, it doesn't have time to change. Whereas in CF, just like many chronic infections, one isolate does not reflect the diversity of the whole infecting population, because over years and years of infection, it's had time to change in very, very different ways. So it's they're very diverse infecting populations. And choosing one isolate is not a good reflection of that diversity. Increasingly, as we heard yesterday from Drs. Rower and Conrad and others, Dr. Kobian, we now know that there are also diverse species infecting people, not just the standard pathogens, but lots of other microbes that our methods just are designed to select against. And so that's two levels of diversity. And the third is that, as I mentioned, there are these very diverse areas within the infected airway where places, for example, have low oxygen, some have high oxygen. And that kind of diversity really hampers how antibiotics are going to work. So that's three levels of diversity. Now, before I move on, I just want to talk again about this standard specimen that we use to sample these infections, and it's what sputum is. This is a problem in this field and for respiratory infections in general. And thinking about what sputum is, I asked a friend, Siri, but not the Siri who's been introducing us, this is the one who lives in my phone, what sputum is defined as. And she told me kindly that it's a mixture of all of the secretions throughout the airways, including saliva and mucus. And we use it in the laboratory, and as it comes up from the lungs, as people expectorate, it of course comes through all of these airways, picks up some bugs, and it also picks up some bugs as it comes through the throat, which has its own airway, or its own bacteria, and through the mouth and the teeth, all of which have their own bacteria. And so that means that the bacteria that we have in sputum may not all have come from the lungs. It does not necessarily completely reflect the infection that we're trying to treat. But for today's talk, we are agnostic to this concept. We didn't set out to answer where the bugs came from, but we decided to start refining the method I'm going to tell you about just by studying sputum, regardless of what it reflects. And so to summarize what I've shown you, traditional methods select for specific bacteria and against some, we select only one or a few isolates of those specific pathogens. And that clearly does not accurately reflect the diversity of microbes that's really infecting somebody, at least early in disease. And so we know then for CF, and I will tell you from lots of other chronic infections, like wound infections, for example, sinus infections, classic laboratory methods do not accurately reflect what's in them. Some of the bugs aren't detected. Only one representative of each bug is detected and tested. It doesn't reflect their behavior, and I didn't talk about this much, but for example, they grow very differently in a test tube, in a liquid, in isolation, where they can't interact with other microbes, and these characteristics really change their behaviors. So do these radiants and nutrients and oxygen and being trapped in a gel. And these tests don't really reflect how these communities respond to antibiotics. They just smolder, despite our hitting them hard with antibiotics, which work somewhat, but not as well as we'd like. So to deal with this, our idea, and the idea that CFRI kindly supported, was to try to create a laboratory test that would allow all of the microbes in sputum, the whole sputum microbial community, to survive, and then test the behaviors, including antibiotic susceptibilities, of that intact sputum microbial community together. And we call that community level testing, which is a concept that's been around in environmental microbiology for some time. But specifically, we are testing antibiotic susceptibility, so we call it community level susceptibility testing, or CLIST. So the method we are developing tests the response of the intact sputum community of bacteria, retaining all the levels of diversity that I talked about, where microbes can interact with each other, using conditions that we hope are closer to what you actually find in CF secretions, including a lot of nutrients that allow all of the microbes we identify to grow and thrive. And we incubate under a range of oxygen tensions, again, to try to reflect that kind of diversity in secretions. So to schematize what we do is we take sputum from our patients. We then mix it in the medium that we're developing, and we're pretty close. I'll show you the results in a minute. We array it into 96 well plastic plates and add increasing concentrations of specific antibiotics that we use to treat patients. We then incubate those plates. We have duplicate plates that we incubate under various oxygen conditions. And then we have a couple of different readouts. And here is the really simple one that is rather powerful. It's really simple. So in this plate, you can see these buttons of bacteria that have grown under these conditions where antibiotics were added at increasing concentrations to the right. Here's a control where you see the bacteria grow in all of these. There's no antibiotic in this row. But in this row, here's an antibiotic, tobramycin, which I'll talk about a lot today, that inhibited all of the bacteria in the community. So we call this the community susceptibility. But as you might imagine, not all the bacteria in this community are necessarily bad. We might want some to stay. So we also want to know at higher resolution which species are being inhibited and which ones are not at these lower concentrations. So and we also want to be able to compare what happens in the test tube to what happened in the source patient when they got the same antibiotic to see if this is really accurate in reflecting what happens in patients. That's the in vivo response compared to the in vitro or laboratory response. So to do that, we use sequencing, as we heard about from Drs. Rower, Kobian, and Conrad yesterday, where we take the original pre-treatment sputum sample from these patients and sequence it and use cultures. And then we get these kinds of graphs that show us each color represents the relative abundance. The size of this box shows you the relative abundance of each of the species that we detect in that sputum sample. And that defines the starting sputum community. We then also can take our CLIST tests and do the same thing. We can sequence those and see what happened to that sputum community when we exposed it to oxygen, different oxygen and antibiotics, and compare the two. See how the sputum community changes in vitro with antibiotics. So that's what we call the in vitro community response to these antibiotics. And our goal in this study is to compare what happened using the most common antibiotic used to treat CF patients in the test tube when we exposed it to tobramycin with what actually happened in that same source patient when they got tobramycin. We want to know if what happened in the test tube is similar to what happened in the patient. So we're comparing the in vitro response to tobramycin with the in vivo response. And for the test set, we use samples from a really exciting study that just was published literally a couple of days ago in Thorax here, which we call the tobramycin inhaled powder study, the TIP study, in which we studied 30 people with CF who kindly consented to give us their sputum, and also agreed to be off of any antibiotics for a month before taking a week of standard maintenance inhaled tobramycin. And then we collected sputum from these patients who came from 3 different centers around the country. We collected sputum from them before, every week during the tobramycin, and then a month after stopping the tobramycin. And we sequenced them. So first we defined the starting microbial communities for all 30 patients before they had any antibiotics and having been off for a month. And this is a complicated slide, no need to focus on details, but these are the 30 starting communities. So the relative abundance of each of these different species, although I'm listing them at the genus level here, just to make it easier, the relative abundance of all of these bacteria are shown here for each patient. Each bar is a patient. And the point I wanted to make is that they're very diverse. The starting microbial communities are really different between people, as we heard about yesterday from Dr. Rower. And a couple of more points I wanted to make are that These green guys and these blue guys are the two bacteria that we generally focus on the most. These are Staphylococcus and Pseudomonas. And you can see that they really vary a lot between patients in terms of how much they're relative, how much they contribute to the community. And in fact, tobramycin is really only prescribed to people who culture Pseudomonas. So this is just an example of how some people have really very little and a lot of other bacteria. And we may have assumed that this is the only bug that was really making this patient sick. So then we did the same thing. We sequenced the sputum samples and used culture as well from the patients during and after using tobramycin. Here is the horrible slide that I don't want you to look at that shows the responses of each patient. Each one of these groups of bars is another patient. But here's a much easier way to look at it. This is a really simple two-dimensional graph called a principal components analysis, where each dot represents the sputum microbial community from a different sputum sample. And they're colored according to what week on the study they were collected. So here are the two data points, the time points where the patients were off of tobramycin. So this is before and after using tobramycin here in red and yellow. And these big dots, by the way, represent the community, the averages among all patients for the two off tobramycin time points. And these are the four on tobramycin time points, really similar, so they cluster together. And the main point is that there's a big change when people are on tobramycin in their sputum communities compared to being off. And all of that change really happens by the first week. So we can really just look at before and 1 week in order to look at the changes. And here's an example in the next slide of what those changes were in terms of bacterial species. And I'm here on the right, just for comparison, I'm showing you the two standard pathogens that we focus the most on, Staphylococcus and Pseudomonas. And on the y-axis is absolute amount of that bacterium before, that's red, being on tobramycin, and blue being on tobramycin, okay? And the main point here is that tobramycin did have some effect on these standard pathogens on average. This is for all patients in vivo, but not even nearly as much as many other taxa, which were just as abundant as those standard pathogens. There were a lot of taxa, and these are generally not bacteria that we culture in our standard methods that were really affected by tobramycin. And there was really not a very big effect on the intended target, which is Pseudomonas aeruginosa. So that was a surprise to us. And I should point out that another paper came out at exactly the same time in the same journal that had the same results. So we're pretty confident in this. So now we've defined the in vivo response to tobramycin. And by the way, I only have a few more slides. I hope I will make it in time. Now we wanted to define what happened in the test tube when we took the starting sputum from those source patients, mixed it with our medium, and looked at tobramycin response at different oxygen tensions. And last year I talked about really refining this method, making the medium, figuring out how we inoculate, and then figuring out how we focus mostly on the live bacteria by getting rid of the dead cells. And in the last year, we've been hampered by coronavirus, as you might imagine, for the last 5 months or so. But we have been able to apply this method and refine it a little bit more to several sputum samples from these 30 patients. And the results are very encouraging. So what I'm showing you here, these are PCR results that show you the absolute abundances on the y-axis, for example, of all bacteria in black with increasing concentrations of tobramycin here. So here's where you see all bacteria are really starting to disappear at this very high concentration. And then here are just 4 different bacteria to show you what contributes the most to this change. And it's Streptococcus in blue, and second Staphylococcus here, and a lot less Pseudomonas, which really seems to be quite responsive to tobramycin. So the big drivers here are 2 taxa that this drug is not intended to target. And this happened not just in the test tube, but even from this source patient, the same was true in the person. So the big changes were from Streptococcus in red, or pink, and Staphylococcus in green. So there are some similarities here, and we've had similar results from all of the samples we've tested so far at all oxygen tensions, which is great. So with my time up, I will just say that we've created a medium that allows a lot of microbes to grow. Our conditions are pretty good. We get similar results under different oxygen tensions. Our analytical pipeline allows us to figure out what concentration of antibiotic inhibits all bacteria and specific bacteria, and how the entire intact community reacts to antibiotics. So for example, how Pseudomonas reacts to that antibiotic when it's surrounded by other bacteria, as it is in vivo. And luckily, I'm glad to say, these results resemble those in the sputum. But one final point, it ain't perfect. Unfortunately, some bacteria still don't grow as well as we'd like in this medium, so we have more work to do, because one of those bacteria is Pseudomonas. It doesn't grow as well as it really should in these conditions. So in the next year, even though our funding is done, because we're excited about this, we're going to keep going and refine this method and try to get to a point where what happens in the test tube reflects what happened in the source patients. And when it does, we'll design a clinical study to see if it leads to better outcomes in patients when we pick antibiotics for them. So I'll just skip this. We're trying to make people better. And I urge you to watch this space. And so I'll end by saying thank you very much, particularly to CFRI. A lot of people contributed to this idea and refining it. And here in the pre-masking era is my laboratory and the 3 very talented scientists who did all of the work that you saw today.